Variable addition and subtraction of nucleotides at the junctions between gene segments contributes to diversity in the third hypervariable region. Of the three hypervariable loops in the protein chains of immunoglobulins, two are encoded within the V gene segment DNA. The third falls at the joint between the V gene segment and the J gene segment, and in the heavy chain it is partially encoded by the D gene segment. In both heavy and light chains, the diversity of CDR3 is significantly increased by the addition and deletion of nucleotides at two steps in the formation of the junctions between gene segments. The added nucleotides are known as P nucleotides and N nucleotides. P nucleotides are so called because they make up palindromic sequences added to the ends of the gene segments. After the formation of the DNA hairpins, the RAG protein complex catalyzes a single-stranded cleavage at a random point within the coding sequence, but near the original point at which the hairpin was first formed. When this cleavage occurs at a different point from the initial break, a single-stranded tail is formed from a few nucleotides of the coding sequence plus the complementary nucleotides from the other DNA strand. In most light chain gene rearrangements, DNA repair enzymes then fill in complementary nucleotides on the single-stranded tails, which would leave short palindromic sequences at the joint if the ends are rejoined without any further exonuclease activity. In heavy chain gene rearrangements, and in some human light chain genes, however, N nucleotides are first added by a quite different mechanism. N nucleotides are so called because they are non-template encoded. They are added by the enzyme terminal deoxynucleotidyl transferase, TDT, to single-stranded ends of the coding DNA after hairpin cleavage. After the addition of up to 20 nucleotides by this enzyme, the two single-stranded stretches at the ends of the gene segments form base pairs over a short region. Repair enzymes then trim off any non-matching bases, synthesize complementary bases to fill in the remaining single-stranded DNA, and ligate it to the P nucleotides. N nucleotides are found especially in the VD and DJ junctions of the assembled heavy chain gene. They are less common in light chain genes because TDT is expressed for only a short period in B cell development during the assembly of the heavy chain gene, which occurs before that of the light chain gene. Nucleotides can also be deleted at gene segment junctions. This is accomplished by as yet unidentified exonucleases. Thus, the length of the heavy chain, CDR3, can be even shorter than the smallest D segment. In some instances, it is difficult, if not impossible, to recognize the D segment that contributed to the CDR3 formation because of the excision of most of its nucleotides. Deletions may also erase the traces of P nucleotide palindromes introduced at the time of hairpin opening. For this reason, many completed VDJ joins do not show obvious evidence of P nucleotides. And that's variable addition and subtraction of nucleotides at the junctions between gene segments contributes to diversity in the third hypervariable region. Have a great day.